All right. Thank you, everyone, for joining us today. Welcome to today's webinar, Enteric Dialysis, No Longer Fiction But a Reality, brought to you by integrativepractitioner.com, and today's webinar is sponsored by Kibow Biotech. I've got just a few housekeeping items before we get started, and I'll go over those as people keep starting to trickle in here. If you have any technical questions throughout the webinar, please use the chat box and I'll respond right away. If you have any issues with your audio today, click on that little phone icon, which is right above the chat window, to receive the teleconference information. To ensure call quality, for those of you that have called in today, everyone's lines have been muted. If you're disconnected, you can log on again using the instructions provided in your initial confirmation email. If you continue to experience difficulty, please email webinar@integrativepractitioner.com. We encourage you to ask questions throughout the presentation today. To type your questions, just simply type your question right into the little Q&A box on the bottom right-hand side of your screen there. And then you should see a send button right there. Hit the send button and select all panelists when you're sending the question, sorry. And please note that we will take all questions at the end of today's webinar. Just due to time, we won't take them throughout. This program will be recorded, and you will receive a thank you email with the on-demand materials within just 2 business days. And at this time, I'd like to hand things over to Usha and Dr. Rangan to get started.
Good afternoon, everyone. Good afternoon, everyone, and welcome to Kibow Biotech's webinar series. This is our last webinar. Earlier, we presented to you 3 webinars. The first was the gut microbiome and dysbiosis in health, disease, and aging. The second one was on kidney disease and obesity. The third was omics everywhere. Today's webinar is based upon our revolutionary concept of using the gut to remove uremic toxins in renal failure. The concept of enteric dialysis, which started as a fiction or a hypothesis, but is now a reality today. Let us look at the different kinds of pathways or metabolic pathways and biochemical pathways what we talk in the human body. In our human body, we have large number of pathways which are running simultaneously for metabolism of carbohydrates, fats, and proteins, for synthesis of energy and for utilization of energy. And from the food that we eat, large number of metabolites which need to be thrown out, what we call as the toxins. All of these are also operated by different pathways in the body. Along with the human pathways, we also have a large number of bacteria that we have on our body and inside our body. And these bacteria also have their own pathways which function in cohesion with our system. And that is why they are very important to us in our well-being and in disease. Let's come first and look at the microbiome. So we know that we have a large number of bacteria on our body and inside our body. So here is where the concept of the microbiome comes in. The microbiome, we define it as the collective genomes or all the genetic material of all the microbes that we have in and on our body parts. They are bacteria, bacteriophages, which infect bacteria, and they're bacterial viruses. There are fungi, there are many protozoans and viruses. And from the study of the Human Microbiome Project, we have learned that there are about 10 times more microbial cells in comparison to the human cells. This human microbiota, or the microbiome as we call, has about 900 to 1,000 different species of microbes. And this makes up extraordinary. It's a diverse collection of different kinds of microbial genomes. Within this human microbiota, which is from different body sites and body parts, we have the human gut alone, which is another site having the maximum number, about 80% of the entire microbiota. And this again is rich in its abundance and its diversity. When about 124 European individuals were studied through the Human Microbiome Project, researchers could isolate something like 3.3 million microbial genes. What does this tell us? So if we compare our human genome and compare the gut microbiome, not the entire microbiome, but just the gut microbiome, then we see that the human genome has about 10 trillion cells. And these code for about 23,000 genes, which carry out various metabolic functions, what we saw in that entire pathway. Now, within this is the human gut, and this gut alone has about 100 trillion cells, and which code for about 3.3 million genes. So what does this tell us? Who is housing whom? Are we humans hosting the microbes, or are the microbes housing us? Because it is they who are larger in number, though they are very, very small and cannot be visualized with the naked eye. So based on this understanding, a new definition came about for the microbiota, and that is that the microbiota can be viewed as a metabolic organ which exquisitely tunes to our physiology and performs functions we have not had to evolve on our own. Taking a step back to understand how this gut microbiome influences us in health and disease. The top 10 leading causes of death in the US is about 2.6 million deaths. And if you see, kidney disease is not the top of the disease list. It is far, far lower. It's on position 9, which will suggest that it is not important. However, that is not so. Kidney disease, though 9th on the list, is very difficult to treat, and it's a very expensive disease. You cannot cure it. There is only ways you can prevent its progression, but you cannot stop it completely. So kidney disease is a very important disease which needs to be focused on. Study of the human genome and the microbiome has now led to an understanding that a large number of diseases are influenced by the gut microbiota. So the gut alone plays an important role in controlling our entire body, starting from the brain, where the gut-brain hypothesis is a recent field which is being worked upon, and it has a role to play in Alzheimer's, Parkinson's, anxiety, and many others. Hypertension, obesity, inflammatory bowel disease, colon cancer, all these are now attributed to the gut microbiota. The chronic kidney disease is also one area where the gut microbiome has a big influence to play. How does this gut microbiome influence us in health, and how do they cause diseases? This is where the balance of the gut microbial ecosystem, as we call it, comes into play. When we see if a healthy human being has a balanced gut microbiota, as we call it, we have a high percentage of the beneficial or the commensal bacteria, which are about 85% in number in the gut. The harmful or the opportunistic pathogens which cause diseases are small in number, and it's about 15%. And since the gut is balanced, there is less permeability of the gut, toxemia or sepsis is low, inflammation is low, insulin sensitivity is high, gut health in terms of metabolic health and cardiovascular health is good. However, what happens is that due to the recent kind of lifestyles that we are leading, where we consume high-fat food, high sugar-containing diets, overeating, a sedentary lifestyle, using antibiotics for various applications, this has led to an imbalance in this ecosystem the ecosystem is wacky, it's out of place. What happens is that more of the harmful pathogens start growing in numbers because they enjoy eating the high-fat, high-sugar diets. They grow in numbers and thereby they repopulate the gut. The beneficial microbes, which are good for health, go down in numbers. And this is a term we called as a gut microbial dysbiosis. This leads to increase in the gut permeability. The intestinal barrier gets inflamed. There is septicemia, endotoxemia. Many of the toxic pathogens make metabolites which are not good for us. And this moves from the gut to your bloodstream, leading to septicemia and toxemia. There is, because of these production of these unwanted metabolites, there is systemic inflammation. Insulin resistance goes up. There is increase in adiposity, diabetes, metabolic syndrome, and all of this which manifest into different kinds of diseases and health problems. So what does this tell? It tells us that it's important for us to have a good balanced microbial system in our gut. And it is possible to get back this balance by having a prudent diet and leading a good lifestyle. And the other option is by taking supplements termed as probiotics and prebiotics, which can restore this gut health. So what are they? Probiotics are nothing but they are the good bacteria or the beneficial bacteria which are important for our gut health. They are mainly fall into two broad groups, that is the lactobacillus and the bifidobacteria. And by definition, they are termed as live microorganisms which, when administered in adequate amounts, confer health benefits on the host. When we say adequate amounts, it is very important because adequate means there's a certain dosage like you have for drugs. It's important that you consume the probiotic bacteria in sufficient numbers, what is stated, to see that health benefit that you want to visualize. If that is not taken in the right amount and the right strain is not ingested, you will not see the health benefit. And this is where the nomenclature of the probiotics come into picture. So when you classify them, what we call as a scientific nomenclature, it is coming as family, genus, species, and strain. So if we take an example of the lactic acid bacteria, the family is Lactobacilluses. Now within this family, there are, for example, two broad genuses. One is the Lactobacillus and the second is the Bifidobacteria. Now within the genus of the Lactobacillus, there are different species. For example, Acidophilus, Plantarum, and Rhamnosus, and many more. Within each of the species of Lactobacillus, again, there are individual strains, and it is that individuality of that strain which gives you that particular health benefits. For example, a Lactobacillus acidophilus strain 123 SAG has a very different property from another Lactobacillus acidophilus strain LA5 or a strain NCFM. Though all three are same Lactobacillus acidophilus, they are three different strains, and so they have three different or maybe more different properties, and each strain does not have the same property as the other. So it is very important that you take the right strain in the right quantity to get the right health benefit. All of these probiotic bacteria are good for health, and they have various therapeutic properties. For example, they have immunomodulatory effects where they strengthen your innate immunity and alleviate food allergies. They normalize your intestinal microbiota composition, giving you a good balanced microbial ecosystem and maintaining gut health. They control IBD. They have importance in antibiotic-associated diarrhea and in traveler's diarrhea. They have various metabolic effects like lowering serum cholesterol, improving the lactose tolerance, and reducing risk of colon cancer. And lately, they have been studied to have neural effects where they have important roles to play in anxiety, in controlling Alzheimer's disease and Parkinson's. Now, for these probiotic bacteria to grow in our gut and to perform the functions what they are supposed to do, they need a healthy food. So that healthy food which the probiotic bacteria need to grow and thrive upon and to show us the benefit, those are called as prebiotics. And by definition, a prebiotic is a fermentable ingredient which allows specific changes both in the composition and the activity in the gastrointestinal microflora that confers benefits upon the well-being of the host and their health. All of these prebiotics are derived from plant sources like raw oats, unrefined barley, wheat, corn, Jerusalem artichokes, and many others. They are all carbohydrates in nature, being of 5 to 10 monomeric hexose carbon units. Nutritionally, we classify them as fibers. All fibers are not prebiotics, but all prebiotics are fibers. Some of the examples include inulin, fructooligosaccharides, galactooligosaccharides, xylooligosaccharides, and arabinogalactans, and also beta-glucans from oats and mushrooms. Probiotics and prebiotics differ in certain aspects. Probiotics are live microbes, whereas prebiotics are non-living fibers. In terms of the heat sensitivity, probiotics are mostly heat sensitive. Since they are live microbes, they have to be refrigerated and stored at the right conditions if they belong to the classes of lactobacillus and the bifidobacteria. Lately, some spore formers belonging to the bacillus group have been classified as probiotics. So if you have a product which has a spore, then you might be able to store it at room temperature and you may not have to refrigerate it. Prebiotics, on the other hand, are fairly heat stable and they can survive low and high temperatures as well. In terms of stability, since probiotics are live, if they are not stored and they being sensitive to so many things, the counts decrease over time. And that count we call as colony forming units, and that decreases over time. So the shelf life is very important for a probiotic. Whereas prebiotics have a long shelf life and they are fairly stable. In terms of the pH, most of the probiotics are supposed to be stable to gastric juice and bile. However, they still exhibit some sensitivity. So it's important that when you take any probiotic product, it has to be encapsulated either as an enteric coated gel cap or an acid resistant gel cap, or the strains of the probiotic bacteria themselves should be coated. Prebiotics, however, are stable at all kinds of pH. I now hand over the presentation to Dr. Rangan, where he will explain more about how pro and prebiotics can be used for chronic kidney failure using our concept of the enteric toxin reduction technology.
Good afternoon, everybody, and thank you very much, Usha, for the great presentation and introduction about probiotics, prebiotics, and gut microbiome. It so happens, incidentally, that we all know that metformin is widely used in the treatment of type 2 diabetes, but its mechanism of action is still poorly understood. Just last May, in Nature Medicine, there was an article which said metformin alters the gut microbiome of individuals with treatment of type 2 diabetic contributes to the therapeutic effects of the drug. Now only we are trying to get some sort of a feel on what we call as gut microbiome. We have now only just started scratching the surface of gut microbiome. It so happened last year, every year the Cleveland Clinic meets in the month of October to predict what are all the technologies that is going to be in the coming year and they predict top 10 technologies. And as you can see, I don't want to go through all the bottom, from top down, but the number one is modulation of the gut microbiome to prevent, diagnose, and treat disease. And if you go, you will see about half a dozen companies in the past 2 or 3 years, which has amassed a large amount of money from various equity and venture capital sources to dig into the gut microbiome. Well, we started a company in 1997, and in about 2 years, there was a symposium that was held at the International Society of Artificial Internal Organs in Edinburgh, Scotland, August 4th, 1999. The topic was, it's a 2-hour symposium. The topic was, will the bowel be the kidney of the future? We did not know what the bowel contains, but we took a bold step to have this symposium. And there are 4 gentlemen there. You can see that extreme right-hand side is Dr. Eli Friedman, a very distinguished nephrologist from Downstate Medical Center. Me, the next one, of course, the tall Indian guy. Next is Dr. Tetsumo Tanaka from Japan, who is a professor of nephrology. And on the extreme left-hand side is Dr. Carl Shellstrand, who is also a distinguished professor of nephrology from the Karolinska Institute. So there are 3 distinguished nephrologists, and I have been assigned the task of taking this concept from a fiction to what is today a sort of a reality. Just to give you some sort of introductory thoughts, you know, we eat various food, and normally we consume proteins, and proteins, the end product of proteins is urea. Of course, you come across uric acid, creatinine. These are all what we call as the metabolites, and we call them as uremic toxins because normally they are eliminated via the urine. So the clean blood with this passes through the kidney, and all the unwanted nitrogenous substances are excreted, and clean blood is returned back to circulation. This is what a normal kidney does. However, when the kidney is impaired, there is an accumulation of urea, uric acid, creatinine, and various other nitrogenous metabolites. And as these toxins accumulate, they promote the growth of what we call as pathogenic bacteria, or pathogenic microbes in the large gut, I should say, and resulting to what we call as dysbiosis. We were originally thinking urea, uric acid, creatinine as a uremic toxin because they are measured in millimolar concentration, whereas we found out that there are over 100 uremic toxins, and some of them present in what we call as a very small concentration called as nanomolar or picomolar concentration, but extremely toxic. And these are normally called as middle molecule, like indoles, phenols, chrysols, parathyroid hormones, etc. And generally, some of these middle molecules are protein bound, and they are not removed by the normal dialysis process. As you go along, you will know, I tell you how we have been successful in trying to address the removal of various uremic toxins. If you take on the right-hand side, you have a healthy population, you have a very high levels of lactobacillus as well as high levels of bifidobacteria. So you are healthy. As Professor said, it's about 85% of good bacteria. You are in a normal, healthy process. And there is in between bacteriocytes, E. coli, enterococci, streptococci. These sort of bacteria, they shift depending upon either side. Not all E. coli is bad. Actually, E. coli is needed, one particular strain, because they synthesize vitamin K, and vitamin K is very important for blood clotting, I should say. Otherwise, we'll bleed to death. Now, in the case of CKD population, what's happening is the accumulation of these uremic toxins promotes the growth of pathogenic bacteria. Now, once they start the pathogenic bacterial growth, there is a reduction of good bacteria, that is the lactobacillus as well as bifidobacteria. Not only this large growth of the pathogenic bacteria also produces unwanted substances due to putrefaction, or what we call as self-fermentation, to produce this what we call as middle molecules, which are totally indoles, phenols, creosols, etc. So basically what we are trying to do is we are trying to actually substantiate by the addition of good bacteria so that we can inhibit the growth or lower the growth of the pathogenic bacteria. So that's the whole concept of it. That is, our concept is restoration of dysbiosis by addition of good bacteria. At this point, I want to digress and give you some sort of an idea. There are 5 stages of kidney failure, stage 1 to stage 5. But by the time we find out that we have kidney failure, we are already on stage 3 or stage 4, because normally the laboratory doesn't report your GFR unless it is lesser than 60. So when you have lesser than 60 to 15 ml, this is what we call as stage 3 and stage 4. And when you have 15 ml, at that particular point, you have no other option other than going in for a transplant or for a dialysis. In USA, you may be surprised there are about 500,000 people who are undergoing either peritoneal or hemodialysis. Hemodialysis about 80%, peritoneal about 20%. Our concept or our idea of starting the company is mainly to delay or postpone the pre-dialysis stage 3, stage 4 patients in reaching stage 5, because that's a humongous savings if we can just do that, as you said. In the case of dialysis patients, we spend close to about $35 billion, because as Usha said, dialysis is one of the most expensive disease. And also, I want to tell you, recently, in the month of May 14, John Oliver had a show on dialysis, and in which he showed absolutely the horrible time that these dialysis patients go through, and the outcome of the dialysis or the quality of life is also not that good. He really opened up a Pandora's box on the dialysis patients and the state of dialysis, in which he also mentioned that U.S. has a universal health care only for dialysis patients, when we spend close to about 1% of our annual GDP on taking care of CKD or dialysis patients, not necessarily of dialysis, because of frequent hospitalization and related costs. And you can go on the website and see about this, what he has opened up and told. Also, you should know that kidney failure is not a primary disease, it is a secondary disease. As Usha said, it is a 9th disease, but although it is the 9th in the listed, but it is the most expensive, because most of the, 1/2 of the diabetic population end up with kidney problem, 1/3 of the hypertension cardiovascular patients end up with kidney problem, the remaining is from HIV, lupus, tuberculosis, etc. So kidney disease is not a primary disease, it is a secondary disease. And if you take kidney, although it's a single organ, it has distinctly 3 distinct functions. One is a secretory function. It secretes what is called as erythropoietin for red blood synthesis, and it secretes vitamin D for calcium as well as thyroxine metabolism, and it secretes renin for the control of blood pressure. It has a regulatory function, regulation of sodium, potassium, phosphate, and trace minerals. That is more or less a homeostasis. And the third important function is elimination of unwanted excretion or elimination of unwanted nitrogenous substances as urea, uric acid, creatinine, and the other nitrogenous metabolites. I will touch back this later, because this is a very important thing that you should know. Although these are 3 different functions, these 3 functions are totally interrelated. Any disturbance in one will affect the 2. And unfortunately, we are now only treating only the symptoms, not the cause. And if you take the differences of the dialysis type, that is their peritoneal dialysis and yet to be, recognized fully, the enteric dialysis, you can see the parameters that in the case of the enteric dialysis patients, this one is very much improved, I should say. And whereas if you take all the peritoneal hemodialysis, it needs surgical procedure, it's invasive, the chance of infection and the dietary restrictions, all these are there. So ours is a very simple concept of using probiotics for the removal of uremic toxins. And the possible modes of action, how the product works, is basically we are introducing probiotics. Probiotics are generally recognized safe by US FDA, and the safety is well assured. And probiotics generally produce what we call as bacteriocin or antimicrobial. These bacteriocin or antimicrobials are more or less small peptide-like molecules having antibiotic-like properties. Since they have antibiotic-like properties, they inhibit the growth of pathogenic microbes. That's the very first thing. Second, it actually has the ability of modulating what we call as immune modulation. In this case, these strains also modulate inflammation in the gut. They lower the levels of pro-inflammatory biomarkers like IL-1 beta and C-reactive protein. C-reactive protein is a collection of almost all biomarkers, and also the middle molecules like IL-6, TNF-alpha, up-regulate the levels of anti-inflammatory markers like IL-10, and also there are some what we call as oxidative stress markers, I should say. They also regulate the oxidative stress markers in a very positive aspect. So thereby, actually, it boosts the immune function of these patients who are highly immune compromised. And the third is, of course, excretive function, excretion of unwanted nitrogenous waste. So also, one of the greatest assets that we have with this, the probiotics is, the probiotics act by these three different mechanisms, but they do not have any drug-drug interactions as of now. And basically, what is happening is, when you take these probiotics, three different strains in a capsule, which used to be enteric coated. Now we have a vegetarian cap. It bypasses the stomach acid because most of the probiotic, when they come in contact with stomach acid, gets killed. It bypasses stomach acid because of the acid resistance, and as it enters the ileocecal portion, if they break open, when they break open, they compete with trillions and trillions of gut microbes they have. So it is not like taking aspirin or something like that, that your headache is going to be over in about a couple of hours or you'll get rid of that. In our case, it may take 4 or 6 or 8 weeks, depending upon patient to patient, for these microbes to compete with trillions of microbes that you have and extricate or utilize the uremic toxins like urea, uric acid as a nutrient. As a nutrient, it eats it or metabolizes it. In that process, what is happening is every 20 minutes there is increased growth, and the increased growth actually takes more and more toxins from the circulating blood around the bowel into the bowel, and the increased growth of microbes are eliminated by the normal defecation process. If you go to our website that is given over there, you will be able to see about, I think it's about 1.5 minutes of video to tell you a video presentation how the product actually works. Well, I should tell you that although our product is a dietary supplement, we have done a pharma-like validation for our product. It is a long, we are a company 20 years old, and when we started, we did an extensive amount of lab work, what we call as research work. We used to take what is called as artificial intestinal fluid, which mimics our gastrointestinal fluid. Into that, we will add urea, uric acid, and creatinine, and some of the nitrogenous waste metabolites. And we will add, the microbes individually or combined together. And periodically we will shake the flask, incubate it at 37 degrees, and periodically every half an hour we will take the aliquot of it to find out how much of uremic toxins is remaining. You will not believe this. We were surprised to find out in about 4 to 6 hours, whatever uremic toxin we just have had totally disappears. And when it totally disappears, the flask to begin with, which is totally transparent, I should say, becomes slightly cloudy, more cloudy, and milky white. And by measuring the optical density, we demonstrated the disappearance of the uremic toxins is directly related to the growth of these microbes. Not only the growth of these microbes, we are actually able to isolate it and enumerate it, identify, yes, these are the growth of the microbes as a result of utilization of uremic toxins. Then we actually found out at Ghent, Belgium, Professor Willy Verstaat and his students were developing a machine. This machine simulates our artificial and gastric intestinal model. It's called a simulated human intestinal microbial ecosystem machine, or SHIME machine. It is a 5-step biochemical reactor. One flask represents stomach, one represents small intestine, one ascending colon, one transfer colon, one descending colon. So these 5 biochemical reactors or flasks are headspace controlled, pH controlled, temperature controlled, even the peristaltic action of the gut controlled. Using this machine, we basically answered 3 important questions. The very first question was, what happens when you put 3 strains of microbes together? Are they fighting each other and killing, or are they hibernating? Second, are they removing the uremic toxins individually and combined together? Third, most important one, that time we did not know even the word gut microbiome. We only know gut as something inside we used to call as gut microbial ecology. Does this disturb the gut microbial ecology? All this we answered in favor by using this machine. Yes, these microbes are still hibernating. They are removing the toxins individually and combined together, and they do not disturb the microbial ecology of the gut. But the most important thing that we also found out was we normally consume about 50 to 60 grams of protein. Most of the end product of the protein is ammonia. We generate about 15 to 25 grams of, urea, 50 to 25 grams of urea. And the urea is normally excreted in a healthy person. In the case of a kidney failure patient, the urea accumulates. So what happens to the urea? So urea is hydrolyzed by one of the strains that we have, which has a very high urease activity, the Streptococcus thermophilus. We are the first one to find out that the Streptococcus urease activity is responsible for the hydrolysis of urea. But what happens when urea is hydrolyzed? It produces ammonia. Ammonia is highly hepatotoxic, I should say. In our case, both in the lab as well as the SHIME machine, we were not able to identify the production of ammonia more than 10 to 20 parts per million. At the same time, if you take urea per se, add a urease enzyme, and put your ammonia and electrode, you see the amount of ammonia produced is 50 to 60,000 parts per million. How do you explain this? Well, I don't know. If you go through your basic biochemistry, you must have read what we call as Krebs cycle or tricarboxyl cycle. They all have alpha keto acid. When an alpha keto acid is aminated by a transamination process, it forms amino acids. Amino acids form tripeptides, polypeptides, and these peptides collectively form the protein structure of the microbe, which is what we see as the increased microbe. So we were thrilled to not only see the microbe, we were able to explain how this growth of microbe happens as a result of the utilization of the transamination reaction of this alpha-keto acid. Well, then the question came, well, those days we had what we call as a 5-6 nephrectomized rat model. You take a rat, remove one kidney and 2/3 of the other kidney, it becomes 5-6 kidney removed. It is 85% kidney function loss. And that particular time, it is stage 5. Normally, they live for about 40-45 days. You may be surprised. In our case, these animals survived for about 145 days. They could have even survived longer. It was a Thanksgiving weekend, and we did this work at Thomas Jefferson University, and we did not have any of the graduate students to take care of this animal, so we actually put all the rats into a CO2 incubator before we went for our turkey dinner. Although we did work with nephrectomized rats, there was a criticism telling me rats' large gut or large bowel does not mimic ours. We went and found out the one which is very close to us is pigs, but the pigs are about 300 to 400 pounds. It is very huge. It's very difficult to handle. So what we did was we were fortunate enough to find out what we call as a mini pig, small pigs, like they are like babies, docile, easy to handle. They are called as Göttingen mini pigs, but that was not available here. It was in Göttingen in Germany. So we imported 2 sets of pigs and we reared them in the Lancaster farm, Lancaster, outside Philadelphia. And as the piglets came after about a couple of weeks, we air transported all of these piglets to Indianapolis, Indiana. There, Professor Willis, as well as his colleague, Dr. Honda, they did the 5-6 nephrectomy, and then they fed the product. After feeding the product, we documented there not the longevity, the level of urea and creatinine comes down. Not only that, these pigs moved so very well, it was very difficult to find out what were the pigs that was fed with placebo and what were the pigs that has been fed with our product. After that, well, we were lucky to find out that cats and dogs also have a kidney failure. There were limited therapies available, and we found out our products has got an excellent impact on cats and dogs with moderate to severe kidney failure. This work was also done. We did some clinical trials on cats, we did some clinical trials on dogs, and we even did some clinical trials on retired zoo animals in Tennessee Zoo. In all this, we demonstrated our products works very well, and it helps to actually get these animals relieved from the kidney problems they had. And we did human clinical trials. We did a multi-site clinical trials in Canada, Argentina, Nigeria, USA. We did a dose escalation studies at 3 and 4 at Thomas Jefferson University. We did what we call as study on dialysis patients at Downstate Medical Center. And the outcome of all these trials is, just to summarize it, up to 270 billion dosing per day was well tolerated. And not only that, there was improved quality of life with respect to physical activity, emotional well-being, as well as a decrease of pain was also reported in some of the patients. And we also found out that the level of inflammatory markers, especially CRP, decreased, and there was a reduction of urea, uric acid, and some of the uremic toxins, including the indoxal glucouronic in the case of dialysis patients. So you can distinctly see this is the outcome from our trials, and we have published all our work. If you go to our website, right from 2001, you will see a large amount of papers and abstracts and poster presentations we have made in various societies, American Society, World Congress, as well as other places. So you can certainly at least agree to me that we have done a very scientific, rigorous study in developing our product like a pharmaceutical-like product. We have also done three. We did even 2013, 2015. We reported a customer survey that the quality of life of these customers who are using our products were very much improved. And recently we completed about 217 survey, and we just have submitted our publications, and I'm pleased to let you know that in all these cases, we have been able to not only stabilize the GFR, but in some cases, even the GFR has increased, I should say. So you will be able to see that as we just go along, publishing our papers. So with 3 consecutive surveys, we have documented that our product helps to improve the quality of life as well as stabilize the restore the dysbiosis state to a stable space as well as improve the GFR. And, there are other products in the market, keto acids. The mechanism is the same thing as I told you, that is transamination to form the amino acids. And, of course, they don't use that in this country. It is quite expensive. And some other countries, they do. Cremazine is a charcoal product. It is used in Japan as a drug. It has been used in Japan as a drug for a long time. It costs about $270. You will have to eat 9 grams of charcoal per day. It is 3 grams per sachet. You have to eat 9 grams. The unfortunate problem with this is the charcoal is used for everything. If you have a poison case, they give you charcoal actually. Charcoal binds everything which is hydrophobic. It does not touch urea at all. And in the case of the patients, it not only removes urea and uric acid and CKD, but a lot of the CKD patients also have a primary disease. So they take some diabetic product, they take some hypertension product. So it helps, actually it hinders, thereby removing some of the products they are taking as drug. In the case of prebiotics, we restore the gut balance and we reduce the production of the toxins, because I told you it is the pathogenic microbes which gives putrefaction products of indole, phenols, etc. So it is much less, and it metabolizes various uremic toxins and is safe, well tolerated. As of now, we have not had any drug-drug interaction or any adverse reaction on the safety of our product. We are now in the process of conducting a large-scale pragmatic clinical trials, both on pre-dialysis as well as a dialysis patient, and document that our product will be universally applicable, and it will be easy to administer, and it will be, although it's a dietary supplement, it is not a drug, because no probiotic as of today is manufactured with a drug master file. They are either what we call as a dietary supplement grade, or they are the food grade. So these are actually dietary supplement. So we only claim helps maintain healthy kidney function. We can't use any of the claims like a drug, therapy, cure, diagnose, etc. So we have the Renadil, 3 distinct products. We deploy 3 different bacteria. One is Streptococcus thermophilus for removal of urea, uric acid, creatinine, and the other one is Lactobacillus acidophilus, removes some creatinine, a large amount of methylamine, dimethylamine, trimethylamine oxide, etc., which is ultimately responsible for the cardiovascular toxicity and cardiovascular death in these kidney failure patients, and it also removes indoles, phenols, cresol, etc., with Bifidobacterium longum. So to sum up, you come across reduction of creatinine, reduction of urea, C-reactive protein, even reduction of potassium to a certain extent, and serum indoxyl sulfate. They're all the uremic toxins. but at the same time increased in quality of life. I always say it doesn't matter what disease you have or what product you take, ultimately what the end result is the quality of life. Is the patient feeling better? Is the patient eating better? Is more productive, more cognitive, less stress on himself or family or caregivers? That is important, and you can distinctly see the quality of life of almost all the patients who have been using our product. 3 out of 4, even other people don't have much problem at all. They feel better. So we have this Azidil, which is a product for cats and dogs, which we have licensed it to a company called Developmental that has been in the market since 2006, and it is one of the hot products that a lot of these veterinary use for cats and dogs with moderate to severe kidney failure. The other one is the Renadil. Renadil is the human product. The human product we sell it only online. If you go to our website, you will be able to order the product online. And as I told you, this is a critical thing which we should know. As we just discussed, a lot of these dialysis patients go through what we call as a yo-yo effect. They go up and down depending upon the date of dialysis or after the date of dialysis. Also, in the case of dialysis patient, because of some of these uremic toxins are protein bound, it is not removed. So they have this yo-yo effect. In our case, what is happening, as I told you, you have got 3 different functions, and all the three different functions, they actually are in a sort of a dynamic equilibrium. So the excretory function will be working 24/7. So to a certain extent, if not completely, it will reestablish what we call as the secretory function as well as the regulatory function. At the present time, the nephrologist treat only the symptoms. If they are anemic, give erythropoietin. If they are having problem with blood pressure, instead of renin, give ACE or ARB inhibitors. And if they are having problem with thyrox and calcium metabolism, give vitamin D. If there is too much of phosphate, give phosphate binders. Too much of potassium, give potassium binders. And to increase the urinary output, they give diuretics, I should say. So we are not only treating the causes, the symptoms, we are actually treating the cause, because as a dynamic equilibrium, 24/7 enteric dialysis will reestablish both the secretory as well as the regulatory function, and that is one of the reasons why we strongly attribute the improved quality of life to all the patients. I was very fortunate to meet the gentleman at the center. His name is Willen Jonathan Kolff. He was the inventor of dialysis machine in 1943. On the right-hand side is Dr. Eli Friedman. This picture was taken about 10 years ago when we had our 10th anniversary. This year we are celebrating our 20th anniversary. Unfortunately, he's no more. But I had the fortune of meeting him not only 10 years ago. I met him in 2002 at Washington in the American Society of Artificial Internal Organs. And casually, as we were chatting, I told him that time when we started our company, our original concept was developing a genetically engineered microbes with various genes like urease gene, uricase gene, creatinase gene, and orally infuse it. As a matter of fact, we got $1 million funding from NADDK to experiment using genetically engineered microbes. But then we just, we had both technical difficulties as well as we know that these patients are highly immune compromised, I should say. And at that time, when he knows that we are doing with the genetic engineered microbes, I told him there is an opposition to that. He said, well, don't worry. Whenever you come up with some sort of a breakthrough or novel ideas, people will always be skeptical. And not only skeptical, they will call you by all their names, I should say. And when I came from Netherlands to Cleveland Clinic, after a few years, when I came up with the idea that we can take the blood out of a CKD patient and clean it up and put it back into circulation, everybody just looked at me like anything and said, my God, what this guy is saying? He must be crazy, lunatic. Send him back to Netherlands. And he said, you won't have that problem I faced, because my first 15 patients died. The 16th one survived for 1 month. Now, I'm sure. You can see the first dialysis machine is probably still in Smithsonian Institute, the size of a washing machine. Now you can see a tabletop machine, I should say. You can even have a machine at home that you can dialysis while you are sleeping, I should say. So the technological improvements have gone so much. So I was delighted to see a sort of a mentor who inspired me to move with what I have been doing in the case of enteric dialysis. So this again, with him, I took the picture, and this is one of the things very interesting that I want to just let you know. When we want to do the clinical trials, our chairman, Dr. Eli Friedman, asked me, well, we want to go and get the permission from the IRB for running clinical trials, and do you have any phase 1 data? I told him, what do you mean by phase 1 data? Our product is a dietary supplement. It is not a drug, I should say. So he said, whether it is a dietary supplement or drug, I need some phase 1 data. I told him, well, Dr. Friedman, we have only at that time we had only the veterinary product, not the human. Then he said, isn't the veterinary human the same? I said, yes. Then I told him, I can't go and afford, to run safety studies with 50 or 100 people. We are a small company. He asked me, can you take the product? I said, yes. How about your wife? My wife was always working with me, who is on the right side. How about employees? We had only 5 employees. Then he said, I need some proof, even if it is half a dozen people, that you have taken the product for 3-4 months and your health is okay, it has not impacted you. So what we did was, our company was, illegal company was Morgan Lewis Baucus, downtown Philadelphia. They drafted an agreement that the employees of Kibow, including the founder and his wife, are all taking the product on their own without coercion of the management or compulsion, and on a voluntary basis. And we, at that time, engaged the services of Professor Nicholas Keflaris. He's no more now. And we went and visited him every month. And we had our physical, we had our medical, we also gave urine, we gave stools as well as blood. It was all sent for outside lab analysis. And for 4 months, all of us were in good health. We had no adverse effect at all, I should say. And Dr. Friedman has got an excellent foresight. He said, Dr. Rangan, make sure you take the first picture before taking your dosage, first dosage, because you guys are really making history. So that is the picture that we took. That was actually published in the World Congress of Nephrology that was held in 2005. The World Congress of Nephrology is held every other year. We have been participating since 2005. Every other year, I think it was followed at Hong Kong, and then it was in Milan, and then it was Rio de Janeiro, then it was in Vancouver, and I think it was in Cape Town, South Africa. About 2 months ago, it was in Mexico. And we also participated since 2005 at all American Society of Nephrology. So now only mostly these nephrologists are opening their eyes, and especially with the idea of the gut microbiome exploding, I should say. We are actually in the cutting edge of exponential advances in treating the chronic kidney disease. Previously, we thought it is urea, uric acid, creatinine. You know that. Urea, uric acid, creatinine. Now we are able to decrease it by using one of our strains, thereby decreasing the growth of the pathogenic microbe. The pathogenic microbe, in turn, actually reduces the putrefaction products like indole, phenol, cresols, or middle molecules. So there is not much of a protein-bound molecules. And by using these 3 microbes, we are basically restoring the gut dysbiosis. Not only we are restoring the gut biosis, we are in a very better position because there is no drug-drug interaction, and it is very safe. And I think the future of chronic kidney disease, both for pre-dialysis as well as dialysis worldwide, will be this most inexpensive dietary supplement, not an expensive drug that you just see outside. And, to make the long story short, I had come across, one of the gentlemen who was the president of the Sai Pharmaceutical when he told me about, what we are doing. And when he mentioned about cremazine, I told him, well, we are trying to develop a product better than cremazine. And he said, do you think you can really do it? I said, that's our hope. Then he asked me, well, Dr. Rangan, how did you come up with the name Kibow? Basically, we came with the name Kibow because we are trying to substitute the failing kidney function by the bowel. That's why it's called Kibow. He said, oh my God, at least you have done one good thing right. If you can prove that your product works better than cremazine, you have no problem in getting into the Japanese market. So I always take pride in telling we not only want to give hope to Japanese kidney failure population of patients, we want to give hope to the entire world population of patients. So certainly, I'm sure you know now, enteric dialysis is not a fiction. It is going to be a reality, and we will be proving it as we just go along with our proposed clinical trials so that it will have a worldwide acceptance and the most economical, costly, least, what do you call as drug effect product on the market. Similarly, there will be other products that will be coming in, and I really want to thank you for your listening, and we would be pleased to take any questions.
Thank you, Dr. Rangan. So we are just about out of time, but I will take just a couple questions here. The first question that came in is, which patient population stage of kidney failure demonstrated the most significant benefit?
The most significant benefit will be those in stage 3 and stage 4. Stage 3 and stage 4. Some people develop rapidly kidney failure because it's very difficult to say. So it is basically targeted mainly towards stage 3 and stage 4.
Thank you. Our next question is, after the initial 3-month dose, what would be a maintenance schedule for patients, and is it stage of disease dependence for maintenance?
We always say, kidney disease is irreversible when once it happens. So it's a lifelong process. Dialysis is a lifelong process, I should say. So would you prefer a 4-hour, every 3 weeks stuck to your chair, or would you like to have just one tablet in the morning, one tablet in the evening issues, So it is maintenance. If you stop it, it will rebound, I should say, because these microbes do not go unlocalized, because there are trillions and trillions of microbes. What you're injecting is only about 90 billion per day.
Thank you, and we'll take just one more question. Can I avoid dialysis by using probiotics?
Say that one more time, please.
Can I avoid dialysis by using probiotics?
No, I cannot say that because it is impossible as a business person, this one, from a regulatory perspective, I'm not supposed to give any advice, etc. But at the same time, we have some case studies, patients, whose GFR was much lower, who have been taking our products feeling better. So I'm sorry, I cannot give any medical advice. No.
Thanks so much. So that is all the time we have for today's program. But if you weren't able to ask your question or you think of any additional questions after this, Dr. Rangan's email is actually on the screen for you now, or you can feel free to email questions to webinar@integrativepractitioner.com. If you could please fill out the evaluation form that will appear once you close out of the webinar today so that we can better serve your needs in future webinars. And as a reminder, the on-demand materials will be emailed to everyone within just 2 business days. I'd like to thank you again, Dr. Rangan and Usha, for a great presentation today, and all of our participants in today's webinar. This webinar was brought to you by integrativepractitioner.com and sponsored by Kibow Biotech. Thank you, everyone, and have a great day.